Welcome, everyone. Today we're diving into the fascinating world of phages with fully synthetic DNA. Researchers from the University of Pittsburgh have pushed boundaries by building bacteriophages gene by gene.
Right. So they're basically turning these viruses into customizable machines against bacteria, correct?
Yep, exactly. It's like building with Lego blocks, where each gene is a block you can add or remove. This lets scientists rewire how these phages operate to, specifically target bacteria involved in infections.
That sounds revolutionary, but isn't it a bit risky tinkering with every gene like that?
Well, scientific progress always has its challenges. Right, and these researchers, led by Graham Hatfull, actually reassembled two naturally occurring phages using synthetic DNA, then experimented by adding and subtracting genes. It's like editing a recipe, where you decide if you need extra spice or want to tone it down.
Yeah. And this could open up new pathways in phage therapy, especially with the increasing threat of antibiotic resistance, huh?
Right on. Absolutely. The ability to design tailor-made phages means we could potentially target, well, specific bacterial strains that are causing infections which conventional antibiotics can't tackle.
Yep, I get that. So essentially, instead of one size fits all, researchers can custom design phages to fight off, say, a resistant bacterial biofilm? Person por. Exactly. It's like having a designer suit. It's tailored exactly to fit the problem at hand. Person 2.
Now, here's something I find intriguing. They aren't just recreating what's found in nature. They are editing even the regulatory genes, the ones that control when a gene turns on or off, which means they can really explore what each gene is doing.
Right, but don't you think playing around with regulatory elements could lead to unpredictable behaviors?
That's a fair point. But
Yeah.
actually, by systematically adding or dropping these genes, they can map out function much more thoroughly. It's like debugging a computer program piece by piece.
And collaborating with companies like ANSA Biotech and New England Biolabs, they bring in top-notch DNA synthesis and assembly techniques. So it's not just theoretical, it's practical, tangible progress.
Right. The synergy between academic insight and biotech innovations is really key here. The implication is huge. It speeds up discovery in phage biology exponentially. Every gene can now be manipulated to ask hard questions like what exactly does this gene do? And what happens if you remove it?
True. So ultimately, this tailored approach could revolutionize our fight against bacterial infections, giving us new tools against a big global challenge.
Exactly. The sky is literally the limit here, as Hatfull put it. It's a fascinating blend of synthetic biology and virology, opening up not just therapeutic possibilities, but also, well, a whole new way to understand viral gene
Indeed, it's both
regulation.
exciting and thought-provoking. I love how this research takes a creative leap into reimagining phages. But I still wonder, don't you think there might be ethical or biosafety challenges to consider?
Absolutely, that's a valid critique. There are definitely risks, and stringent safety protocols are essential as we push the envelope. In any case, this breakthrough represents a significant step in the synthetic biology revolution, one where our imagination really drives scientific innovation.
Couldn't agree more.